Welcome to this session on contamination types in pharmaceuticals. In today's video, we'll explore the risks contamination poses to products, patients, and businesses, and by the end, you'll know the key strategies to prevent and control them. Here's what we'll cover: the role and impact of contamination, the big four contaminants, global rules of the game, 7 prevention pillars, practical tools you can use on the floor, and how to sustain compliance over time. In the second half, we'll connect contamination control to business culture. We'll also talk about the business case and wrap up with next actions and closing thoughts. Contamination is one of the leading causes of recalls in pharma. Each batch failure can cost millions, and the ultimate goal is always zero cross, zero microbe, zero particle. We'll look at real-world case studies, 7 prevention pillars every site should audit, and quick floor-ready tools to keep teams vigilant. From 2020 to 2024, recalls were dominated by cross-contamination at 38%, microbial issues at 31%, and chemical or particulate problems making up the rest. These figures prove systems often fail when tested in reality. A single recall can cost over 55 million euros, drop stock prices by 7% the same day, and even cost executives their jobs. Contamination is both a quality and a boardroom issue. The big four are cross-contamination, microbial, particulate, and chemical. Each one requires different detection and control strategies. Cross-contamination happens when traces of one product carry into another, even at invisible levels. Detecting and preventing it requires sensitive analytical methods and strict facility controls. Shared equipment, poor gowning, and inadequate cleaning are the main root causes. Even small lapses in operator behavior can spread contaminants between batches. The heparin tragedy showed how deadly contamination can be. Standard tests failed to catch it, leading to dozens of patient deaths worldwide. Companies use risk matrices to decide between shared or dedicated facilities. Potency, toxicity, and process cleanability all factor into the decision. Microbial contamination involves bacteria, mold, yeast, and endotoxins. These organisms can grow during shelf life, leading to fever, sepsis, and even death. Microbes thrive in hoses, condensate, standing water, and even on operator gloves. Controlling these hotspots requires vigilance and strict hygiene. The NECC meningitis outbreak was caused by poor cleanroom practices in a compounding pharmacy. It killed 64 people and remains one of Pharma's darkest contamination cases. Particulates include visible fibers, flakes, and rubber particles. They can block blood vessels and are tightly controlled under USP and EP standards. Particles often come from glass vials, rubber stoppers, or equipment abrasion. Even packaging dust can contaminate a product if controls are weak. One major recall involved glass flakes contaminating vials of a biologic product. Over 2 million vials were withdrawn because of an unqualified vial supplier. Chemical contamination comes from solvents, detergents, or degradation products. Even small amounts beyond PDE or TTC limits are unacceptable. Maximum allowable carryover, or MAC, uses therapeutic dose, batch size, and safety factors to set scientific residue limits. It's a calculation, not guesswork. In one case, chlorhexidine from a shared cleaning system contaminated an oral spray. The failure showed why even cleaning agents themselves must be strictly validated. EU GMP requires prevention of cross-contamination and health-based exposure limits for cleaning. Validated procedures are mandatory for any shared equipment. The FDA outlines cleaning, air handling, and contamination prevention in CFR 211. These sections form the baseline of U.S. contamination control. PIC/S adds further rigor, requiring a cleaning validation master plan and risk-based inspections. Sites must be prepared for deeper scrutiny. ICH Q9 introduces risk management, while Q10 establishes a pharmaceutical quality system. Together, they frame contamination control as an ongoing life cycle activity. The first prevention pillar is dedicated equipment. It's mandatory for potent drugs and high-risk products where HBL limits are extremely low. Dedicated equipment is essential for cytotoxics, hormones, and sterile beta-lactams. Regulators expect strong justifications if shared systems are used. The second pillar is validated cleaning, where processes must consistently achieve MAC limits. Worst-case testing across multiple runs proves robustness. Cleaning validation follows a life cycle: design and MAC calculation, qualification, then ongoing verification. This ensures controls remain effective over time. Worst-case products are chosen based on solubility, potency, and batch size. This proves the cleaning process can handle the toughest scenarios. Analytical methods must be specific, sensitive, validated, and rapid. Techniques like TOC or UPLC are often preferred for real-time reliability. The third pillar is SOPs that are clear, visual, and tightly controlled. Vague instructions are one of the most common causes of contamination errors. Training goes beyond reading and signing documents. Operators must be observed, tested, and retrained after any deviation. Good design prevents contamination before it starts. Unidirectional flows, physical barriers, and HVAC cascades are all essential. Grade B areas require 20 air changes per hour, HEPA H14 filters, and no recirculation between products. Air design is a frontline defense. Regular GMP training builds a culture of quality. Simulations and refreshers ensure people stay sharp year after year. 70% of contamination comes from human error. Nudging, reinforcement, and easy-to-follow procedures build better habits. Real-time monitoring has replaced snapshot checks. Modern counters and alarms provide early warnings before risks escalate. Alert and action levels are set using baseline data and statistics. This keeps monitoring scientific, not arbitrary. Rapid microbiological methods cut detection times from days to hours. Faster release reduces quarantine and boosts supply chain efficiency. The seventh pillar is controlled material flow. Inputs, outputs, and waste must never cross paths inside a facility. Spaghetti diagrams often reveal risky personnel movements. Redesigning routes to eliminate intersections lowers contamination risk. Quick 30-second floor checks make contamination control part of daily culture. Operators should know cleaning limits and environmental data at all times. Daily scorecards gamify compliance. A simple green, amber, red system keeps contamination awareness visible. Tablets, QR codes, and dashboards bring transparency to cleaning and monitoring. Digital tools help QA and QC stay connected. Compliance must extend to suppliers. Audits and cleaning data reviews ensure upstream partners don't introduce risks. Changes in detergents, packaging, or equipment all require new validations. Skipping this step has caused many recalls. Root cause analysis tools like 5 Whys and fishbone diagrams reveal the real problems. CAPAs must always address system design, not just symptoms. Effectiveness is proven when repeat deviations drop and contamination events trend downward. If metrics don't improve, the CAPA isn't working. Validation must be maintained through annual reviews and requalification. Continuous process verification feeds back into contamination control. A CCS links all prevention pillars into one framework. It should be approved by QA and embedded in the site master file. Sites must prepare contamination packages for regulators. This includes validation summaries, MAC calculations, and trending reports. Mock audits with cross-functional teams are the best preparation. Using checklists and RAG ratings ensures readiness. Contamination control protects both patients and profits. A strong culture treats prevention as a daily responsibility, not an occasional task. Thank you for joining this deep dive into contamination types in pharmaceuticals. Prevention isn't just compliance, it's a commitment to patient safety and trust.